The Nobel Prize in Physiology or Medicine in 2009 was awarded for how chromosomes are protected by telomeres in the enzyme telomerase. The science is really very simple. Telomeres are our body clock. They shorten with normal cell division. Telomerase is an enzyme that causes the telomeres to lengthen. And a gene to produce telomerase is present in every body cell. And telomerase production can be switched on by substances known as telomerase activators. And bad things happen when the telomeres get short. Dr. Bill Andrews was one of the co-discoverers of human telomerase. He was awarded second place for National Inventor of the Year in 1997 for discovering telomerase. He's been named on 35 US-issued telomerase patents. He's certainly a world-leading biologist in telomere research. He's also a brilliant educator and lecturer. And one of the things that always amazed me was, why would a guy spend his life trying to cure aging? Well, basically it was because as a child, Bill's father had said to him numerous times, "Son, when you grow up, why don't you become a doctor and cure aging?" I'm very grateful that Bill has allowed me to share with you a lecture he presented to a group of anti-aging medical specialists at their 2009 conference. The American Academy of Anti-Aging Medicine represents 26,000 physicians and scientists from 120 countries.
Dr. Andrews leads the scientific research and development function of Sierra Sciences. His team is now pursuing direct molecular approaches to temporarily activate telomerase expression and is performing discovery efforts. While director of the molecular biology at Geron Corporation, he was one of the principal discoverers of the telomerase gene. I'm proud to introduce Dr. William H. Andrews.
Groucho Marx once said, "I intend to live forever or die trying." He was trying to be funny when he said this, and he got a lot of laughs. But I don't believe the people in this room think this is very funny, because we're here because we're all trying to live forever. I know I am. And we are going to try our best to do that. And so what I'm going to be talking today isn't really how to live forever. I'm going to be talking about how to try to live forever. I'm going to be talking about research science that's underway now that's going to allow us or increases our chances of succeeding at this. And so pretty much what I'm going to be talking about today is not what you can buy today. In some cases that's not true. But I'm going to be talking about what's in the future, what we have in the future to look forward to. So how to try to live forever, first do the obvious things. Eat healthy, live healthy, exercise, listen to everything your doctor says. But this isn't enough. This isn't going to allow us to do things like run 7-minute miles when we're 130 years old. And that's what we want to do. And by the way, we're not just talking about living longer, we're talking about living healthy longer. We want to be very healthy when we're in our hundreds. Now, there's been a lot of things that have allowed us to extend our lifespan tremendously, especially just in the last 100 years. In 1900, the average lifespan was only 47.3 years. And since then, we've extended that up to 78 years through a lot of things, including better sewage systems, vaccines, refrigeration, exercise. The other column has coronary artery bypass surgery, chemotherapy. All these things add up to give us a tremendous extension of life. But none of these things are going to cure aging. We still aren't going to see anybody, or we aren't going to be running 7-minute miles when we're 130 years old. And this is because there's a theoretical maximum lifespan that a lot of scientists have calculated to be 125 years. Apparently nobody can live beyond 125 years. There's been at least 3 studies done. References are here. The most recent one was just a few months ago, a mathematical model looking at how much older people have been getting over the last 100 years, and still they see this threshold. We can't seem to pass this threshold of 125 years, and they don't know why. Most of you have probably never heard of a telomere, and so I'm going to give you a basic background on what this is first. And to do this, I need to zoom in on a person. And when we zoom in on a person, we see that a person is made up of 100 trillion cells. And if all those theories I was talking about before on why we age, they all relate to the aging of our cells. We age because our cells age. If we could find a way to prevent aging in our cells, that would probably cure our own aging. If we zoom in even further, we see that every cell contains a nucleus. And inside these nuclei, there are chromosomes. And these chromosomes are where all the genes exist to give us our blue eyes, our blonde hair, make some of us short and make some of us smart. And so I want to zoom in even further now and explain that a chromosome is made up of a DNA strand that extends from one end all the way to the other. And the DNA is approximately 10 million bases long. It's made up of units called bases, and it's pretty much coiled up like a slinky all the way through this chromosome. The key of my talk today is the very tips of the chromosomes. These are what are called the telomeres. And if we unravel this slinky in the telomere, we can see that a telomere is about 15,000 bases long. At least it is when you're first conceived. As soon as cells start dividing, your telomeres start getting shorter. And by the time you have enough cell division to become a newborn baby, the telomeres have shortened already down to 10,000 bases. They've gone from 15,000 bases to 10,000 bases. As cells start to divide further, for us to become adults, our telomeres shorten even further. And when they get as short as 5,000 bases, we die of old age. Telomeres cannot get shorter than 5,000 bases. Inside of petri dishes, when we grow cells in petri dishes, the cells absolutely stop growing and die of old age at 5,000 bases. Let me repeat this. We are born at 15,000 bases. We are conceived at 15,000 bases. We're born at 10,000 bases. We die of old age at 5,000 bases. And there's absolutely nothing we can do about this. This telomere shortening is going to occur no matter how well you exercise, no matter how well you eat, no matter how much you do everything your doctor tells you to do. You cannot stop this telomere shortening. Now, this is the only clock of aging that anybody has ever discovered in humans. And it actually fairly well explains this 125-year theoretical maximum we have, because these telomeres shorten approximately 1,000 bases per year. So this is a new word. A lot of you now know what this word means, but I predict that in the next 5 to 10 years, this is going to become a household word. Everybody's going to want to know how long their telomeres are. I believe this is going to be the biggest and best biomarker of aging and overall health that we've ever seen. So the best example of where telomeres play a role in aging is the fact that there are some people who are born with already short telomeres. This disease, called progeria, affects these kids. And this disease is called the disease of short telomeres. These kids have a maximum life expectancy of about 20 years, and they suffer from all the same age-related ailments that normal old people do. If we could find a way to stop telomere shortening or to slow it down, this could be a cure for this disease. That's pretty much going to be what I'm going to be talking about today, is finding ways to stop this telomere shortening. Now, these kids aren't the only ones that suffer from telomere shortening. We all do. Telomere shortening has been implicated in almost all aspects of normal aging. And what I'd like to do is I'd like to just give a few examples, and I'll start off with the number 1 and 2 causes of death. Cancer. When our telomeres get short, our chromosomes become unstable, and the rearrangements of our chromosomes cause mutations, and these can induce the expression of oncogenes and de-repressed tumor suppressor genes, increasing our risk of getting cancer. In addition, when the telomeres in our immune cells get short, our immune cells lose the ability to fight the cancers. So it's two things happening here. Short telomeres increase our risk of getting cancer and decrease our ability to fight the cancer. Atherosclerosis. When you look at the cells lining your blood vessels, the endothelial cells and the smooth muscle cells, you'll find that if you look at the telomeres in the cells surrounding plaque, you'll find those telomeres are shorter than the telomeres in other parts of the blood vessels. And this is because we think that the the cells have gotten older through telomere shortening, and as a result, they are less capable of preventing plaque. So if we could find a way to prevent that telomere shortening, this could be a possible way of preventing plaque and even fighting plaque. There's a lot of disease, lack of time. I'm not going to go into all of them, but people are welcome to talk to me throughout this conference. I can tell you the basics of telomere biology in all of these, but they include Alzheimer's, osteoarthritis, osteoporosis, just general skin aging. They include macular degeneration, which is the #1 cause of blindness in the elderly, liver cirrhosis, muscular dystrophy, AIDS. Now, if you all remember back in the early 80s when AIDS epidemic first came around, doctors were saying that they don't understand why their patients don't have any T cells. But we now know that the immune cells, the T cells in the immune system, put up such a fight against the AIDS virus, and the fight was unsuccessful. But because of this fight, there was lots of cell division, and the telomeres got very short in these immune cells, and the immune system died of old age. And that's why the AIDS patients don't have T cells. Now, if we could find a way to prevent the telomere shortening in these immune cells, the immune cells would stay around. They probably wouldn't be successful at fighting the disease, but the person might be able to live a normal life. General immunity, stem cells. Every time I go to a stem cell conference, doctors are asking me, have we come up with a drug to prevent telomere shortening yet? And I always have to say, not yet. But the reason why they're asking is because when you take stem cells out of a person, you have to expand them in culture. You have to get more numbers of them. You have to let them divide. Well, because of the cell division, the telomeres are getting shorter, and so the cells that they put back into a person end up actually being older than the cells they took out of a person. So the people doing stem cells need a drug that will prevent telomere shortening when they're doing stem cell therapy. There's a lot of diseases. Anything you can think about that involves cell division is going to have telomere shortening. And one thing I want to leave you with is that bad things happen when telomeres get short. There's nothing good about short telomeres. And there's also nothing bad about long telomeres. So it would be good if we could find a way to keep the tips of our chromosomes long. So now let me address the question of why do telomeres shorten? And to do this, I have to explain, when I was showing you the cell and it had the chromosomes in it, when that cell divides, all the chromosomes, the two new cells that get made out of this have to have all the same chromosomes that the original cell had. So the DNA has to duplicate. It has to replicate. Now, DNA replication is a lot like laying bricks on a brick wall. The bottom strand here can be considered the original strand, and then the new strand, the top layer is the new strand, and this has to be identical to the original strand. Remember, this DNA is about 100 million bases long, so it's a long, arduous process. And if there's any mutations that are made during this thing, or any mistakes, this can lead to mutations that can cause cancer, etc. So this is a pretty accurate procedure. But what we want to do is we want to look what's happening over at the very tips of the chromosome, okay, the telomeres. And so we have this brick layer, which, by the way, in a cell is called DNA polymerase I, is moving along, replicating, and you're going to see there's a problem coming up here. And this is what actually happens inside of a cell. The cell is unable to replicate the very end of the chromosome. As a result, every time DNA or chromosomes are replicated, they are a little bit shorter. And as I said before, there's nothing we can do about this. But I want to point out that there was no degradation here. There was no enzymes that came along and chewed this away. This is purely due to a lack of an ability for the cell to replicate the very ends of the chromosome. It's more of a passive, not an active phenomenon. But now this DNA has to divide, or the cell has to divide, the chromosome needs to replicate again. The new chromosome is even shorter, and again and again. And there's absolutely nothing we can do about this. No matter how well you eat, no matter how much you exercise, no matter how much you do, everything your doctor tells you to do, you cannot stop this telomere shortening. I said you can't stop it, but there are things you can do to accelerate this telomere shortening. And this has to do with anything related to an unhealthy lifestyle. And that includes things like obesity, lack of exercise, psychological distress, and smoking. Things like those will create free radicals inside your cells that will come along, and the free radicals will clip your telomeres and make them shorter. Now, this kind of stuff you can control. You've probably seen press releases lately where people are saying that simple meditation can help reduce telomere shortening. Well, this is why. It's reducing the accelerated telomere shortening, but there's absolutely nothing that will work to actually reduce this basal level of telomere shortening here that I described first. So, how are we going to stop this? I've been saying that there's nothing you can do about this, but that's all I'm trying to do, is I'm trying to figure out a way to do this. And I've learned from a lesson from our reproductive cells. Our reproductive cells do not age. And this is important because if they did age, our children would be born as old as we are. But they don't age, and that's why our children are born young. So something inside our reproductive cells prevents them from aging. And so this was a key to try to find out how to make our other cells not age. Well, as it turns out, our reproductive cells do not have telomere shortening, and this is probably why they don't age. And this is because, we discovered this about 15 years ago, our reproductive cells contain an enzyme called telomerase. And this is a picture of a cartoon of telomerase. The green is the DNA, showing the very telomere tip of the DNA. And the factory-looking thing is the telomerase enzyme. And what it's doing there is it's lengthening the telomere. Now, so let's go back to this original model here of the brick layer as an analogy for DNA replication. This brick layer is still going to fall off in a reproductive cell. But like an angel, telomerase comes in and replaces that brick, or fills in the missing DNA strand. So as a result, our telomeres don't get shorter. And if you think about it, they can't be getting shorter because you came from cells from your parents, they came from cells from your grandparents, et cetera, et cetera. Your children are going to come from cells from you. And if we had telomere shortening all the way through here, you would never, ever be alive right now because you wouldn't have any DNA left. There'd be so much telomere shortening. So our simple strategy in my labs is to take into account that first, somatic cells don't produce telomerase. That's the problem. But they do contain the gene for telomerase, so which means they have the ability to produce telomerase, they just don't. And they don't because the gene is shut off. And so our simple strategy, obviously, is let's figure out a way to turn it back on again. So inside the reproductive cell, telomerase is produced by the telomerase gene. And next to every gene, there's a regulatory element that's like a light switch that turns the gene on or off. In this case, in the reproductive cells, the gene is turned on. So in all of our other cells, a repressor comes along and binds to that regulatory element and shuts the gene off. And that's called a repressor. So what we want to do in my labs is we want to find a drug that will bind to that repressor, remove that repressor from the regulatory element, and allow telomerase to turn back on. If you decide you don't want telomerase to stay on forever, you can quit taking the drug and the repressor will go back and shut it off again. So that drug would be considered an anti-aging drug. This is the team working in my labs, where there's about 30 scientists. We have a 10,000 square foot research facility in Reno, Nevada. We've been in operation now for 10 years. And we have $2 million robots that are spending all their time just screening drugs after drugs after drugs. And you can see like a robotic arm right there that will carry like 96 different drugs at a time, carry from station to station, and analyze, have equipment on this robot that will analyze the weather, see whether telomerase turned on. The key is, I want everybody to realize there's a very significant effort going on here to try to cure aging. And I have to admit, one of the main reasons this is going on is because I want to cure my aging. I'm very, very motivated by that. So we've been in operation for 10 years, and as of just a week ago, we have screened 141,756 chemicals, and we have now found 32 hits. That is, we have found 32 of these green things that dislodge this repressor. That is 32 different chemicals that turn on the telomerase gene. But this could be the biggest thing that ever hit the planet. Telomerase induction might just actually change the way all of us live if it works the way we think it's going to work. Now, this is probably where I should end my talk, because this is where we're at right now. But I want to say that we don't know the answer to this question yet. Will telomerase induction cure aging? It's all a theory right now. It's all a hypothesis. And so we don't know the answer to this. But I want to say that there is a light at the end of this tunnel here. There is a lot of reason for a lot of hope. And that's because of proof of concept experiments that we have done over the years. Now, ideally, as I've been saying, we'd like to find a drug that would turn on the telomerase gene by just dislodging that repressor, but that drug doesn't exist yet. And so what we've done in the past is we've introduced the telomerase gene through a process that we call telomerizing. We take the gene, we insert it into the chromosome by carrying it on a virus, and then we do it in such a way that it's not regulated, and so that gene is producing telomerase. And so we've done that, and first let me say that when you take a normal cell, a normal skin cell, and grow it in culture, the x-axis here is the number of days in culture, and the y-axis is the number of cell divisions, you find that after a time the cells start slowing down and level off. This is called cell senescence, and that leveling off place is called the Hayflick limit. This has been true for all cells ever, but for the first time ever, when we telomerized human skin cells in 1997, we found that they did not level off. They kept on growing. Nobody had ever seen this before. These cells were the healthiest cells in the world, and they also had a tremendously decreased risk of getting mutations, including cancer. So we published this in 1998, made headline news all over the world, and it really opened the door for a lot more research on telomerase. But one big surprise in this study is that the telomeres didn't just stop shortening, as we expected. They actually got longer. So now this begged the question, did we actually make the cells younger? And this was not really our goal at the time, but let's take it. Now, the most exciting proof of concept is very recent data. This is proof of concept actually in humans. There actually is a commercially available nutraceutical that people can buy now for use in humans. And the company that's making this nutraceutical has provided us with some of it to do research on. And we have added it to cells, and we have found that it in fact induces telomerase expression. Not very much, but little is better than nothing. Even a little bit will at least decrease the rate of shortening. But now, it might even be working better than we thought. In collaboration with another lab, we took blood samples from people that are actually taking this nutraceutical now and looked at their telomeres. And even though we couldn't see the average telomere increasing, we could see that the shortest telomeres were increasing, which is telling us that this nutraceutical is inducing telomerase, and this telomerase is having an effect on the telomeres. When people that had been on the nutraceutical for more than a year were looked at, there were several different things that were seen. Some of these biomarkers a lot of you are familiar with, but CD28 minus T cells is one of the best biomarkers of aging, and they tend to increase when you get older. But the people taking this nutraceutical saw their T cells, CD28 negative T cells, decrease. They also saw naive T cells increase, natural killer cells decrease, blood pressure decrease, skin elasticity increase. This is really exciting. This is a real proof of concept that telomerase induction is something that can really be used to potentially cure aging. Now, we still don't know the question, will telomerase induction cure aging? And that's because this new nutraceutical hasn't been around long enough to really answer the question. But things are looking good, and I'm really looking forward to seeing what happens. We also still don't know the answer to how long we will live if we induce telomerase. But I predict that people will definitely live beyond the 125-year theoretical maximum. So, in conclusion, I'd like to say that Groucho Marx may not have been joking. Maybe he really did want to try to live forever. I know that I do, and I know that my company is very motivated to doing this. Our logo is Cure Aging or Die Trying.
Even though what Bill shared back in 2009 was exciting, since then even more dramatic proof-of-concept experiments have been published by scientists from Harvard Medical School. But for me, the most critical experiment was the experiment that Bill Andrews and his team had done way back in 1997. Because for me, this experiment showed what happens to cells in perfect conditions. Dr. Leonard Hayflick had discovered way back in 1961 that a population of normal human fetal cells in a culture containing all of the essential nutrients will only divide somewhere between 40 and 60 times. Now, these are cells in perfect conditions. They've got all the nutrients they need. No oxidative stress, no psychological stress, no toxins. Just cells growing perfectly in a culture. Because what they found was that the telomeres just shorten with each cell division. And when they get down to 5,000 units long, the cells die. And adding more resveratrol, CoQ10, fruit extracts, antioxidants, glutathione, vitamins, colostrum, stem cells, or treated water makes no difference. These cells have already got all the nutrients they need. The only thing that had ever been shown to affect the Hayflick limit was when the cells were telomerized. And well, when the cells were telomerized, functionally they became immortal. They just kept dividing and basically until they filled the Petri dish. Forever. And this work was published by Dr. Bill Andrews and his team back in 1998. And when they looked at the telomeres, what they found was that the cells didn't just stop shortening, but they actually got longer. And this caused the scientists of the day to ask the question, does this mean that the cells had got younger? Well, really, that question was answered definitively by a group of scientists from Harvard Medical School led by Dr. Ronald DePino when they published on the 28th of November 2010 in the peer-reviewed journal Nature research demonstrating for the first time that turning on telomerase and lengthening telomeres will reverse the symptoms of old age in a living animal. It's important to understand what they've said here. They didn't say they would slow it down. They didn't say it would stop it. What they said was that it actually reversed the symptoms of old age in a living animal.
And now, eternal youth. Is it in a cage around the corner? News tonight of a breakthrough for some pioneering mice. But we always wonder, what does a fountain of youth for rodents reveal for humans? Here's Sharon Alfonsi reporting.
I feel tremendous.
In the movie Cocoon, it's a swimming pool that turns back the clock for a group of senior citizens. But now researchers have found a way not just to stop, but reverse the aging process. The key is something called a telomere. We all have them. They're the tips or caps of your chromosomes, seen here in yellow. This is what it looks like in a young adult. But as you grow older, the telomeres become damaged and frayed, and as they stop working, we start aging, experiencing things like hearing and memory loss. Scientists took mice who were prematurely aged, added an enzyme, and essentially turned their telomeres back on. You can see it before the enzyme, after.
Their brain function improved, their fertility was restored. It was a remarkable reversal of the aging process.
Look at this picture. The mouse on the right has bad skin, gray hair, and is balding, but the one on the left had its telomeres flipped back on.
And you could see that essentially you now have a dark coat color, that the hair is restored, that the coat has a nice healthy sheen to it.
Even more dramatic, the change in brain size. This is before. The mice had 75% of a normal brain, like a patient with severe Alzheimer's. But after the telomeres were reactivated, the brain returns to normal size. As for humans, while it is just one factor, scientists now say by looking at our blood cells and measuring those telomeres, you can get a better idea of how well you'll age. The longer the telomere, the better the chances for a more graceful aging.
Later, Dr. Ronald DePino stated, these results prove that aged tissues have a point of return and can regenerate if critically short telomeres are lengthened. And Dr. Bill Andrews said, these were results that, if seen in elderly humans, would constitute a successful reversal of the aging process. And he said, just as telomerase activation reversed degeneration in the organs of these mice, we expect that it can do the same for humans. As best scientists can tell, lobsters show no measurable signs of aging. They have no loss of appetite, no change in metabolism, no loss of reproductive urge or ability, and no decline in strength or health. I mean, how good would that be? Lobsters, when they die, they seem to die from external causes. They get fished by humans, eaten by seals, wasted by parasites, but they don't seem to die from within. Lobsters possess the fountain of youth. And in humans, telomerase is only found in some types of tissues. But in lobsters, telomerase is found in all types of tissue. And with a steady, evenly distributed supply of telomerase, lobsters don't approach the Hayflick limit, which means their cells stay pristine, young, and dividing. And that's why a big old lobster in a restaurant tastes just as good as a smaller one. And that doesn't happen with beef, or lamb, or chicken. You see, lobsters don't age. And what scientists have also found is that lobsters don't get cancer. I mean, what scientists believe is their immune system stays functional and healthy for their entire life. I can't tell you how excited I was when I found out about lobsters and realised that God had already created one animal in which the gene to produce telomerase had been turned on in every cell in its body, and that animal was doing extremely well. Dogs, cats, and horses also age by telomere shortening. So to summarise, the science is really very simple. Telomeres are our body clock. They shorten with normal cell division. Telomerase is an enzyme that causes the telomeres to lengthen. And a gene to produce telomerase is present in every body cell. And telomerase production can be switched on by substances known as telomerase activators. And bad things happen when the telomeres get short.